Today we are joined by Dr. Lindsey Hoffman, a postdoctoral researcher working with Dr. Stacy Bonos1, also a NEWBio contributor, in the Department of Plant Biology at Rutgers University. Dr. Hoffman received her PhD in 2013 from the Stockbridge School of Agriculture at the University of Massachusetts Amherst. Her research focused on abiotic stress physiology of grasses, and her dissertation work investigated the freezing tolerance mechanisms of temperate perennial grass species. She's currently funded by NEWBio and is working with Dr. Bonos1 on breeding switchgrass for improved yield and disease resistance on marginal lands. So she'll be sharing some of her thoughts on this work with us today. Lindsey, welcome.
Hi, thank you for that introduction, Sarah. Again, please, if you're having trouble hearing me at all, just feel free to type in the chat box and let me know. We can work on fixing that audio. So, as Sarah had mentioned, I am a postdoc and I'm currently working on my third year with Dr. Bonos1 at Rutgers University. And the primary two projects that we've been working on through the NEWBio, the Northeast Woody Warm Season Biomass Initiative, is improving switchgrass for both marginal growth and disease tolerance. And so before we get into the actual projects that I'm working on, again, I just wanted to mention that the grant as a whole is a collaborative project that involves 8 universities and 4 national organizations. And so within that initiative, it's integrating several areas of research, looking at growing biomass and then how to use that for the future. And in particular, within the Switchgrass Feedstock Improvement Division, Rutgers, Cornell, and Penn State are working collaboratively together on the breeding efforts. So just as a brief review and introduction for those of you who may not be familiar with switchgrass, it is a warm season perennial bunch grass, which makes it a nice choice for bioenergy purposes because it is a perennial. The species consists of 2 different ecotypes. So there are upland and lowland ecotypes. And those ecotypes allow the species to survive in many diverse environmental and climatic conditions, which is nice to have a wide growing range to be able to produce the species for bioenergy purposes. And so differences both at the phenotype and the genotype level, things associated with abiotic stress tolerance, for example, allow, let's say, upland ecotypes to tolerate drier, colder conditions, where lowlands are more adapted to warmer regions that may be prone to more wetter conditions. And so when we look at the distribution of switchgrass throughout the United States, you can see that although there are definitive lines drawn on this map, that there is some overlap where we can grow lowland ecotypes in the northern parts of the United States, and the upland and lowland ecotypes can also be grown in the transition zone. So with regards to switchgrass research, there has been a lot over the past several decades because switchgrass was named by the United States Department of Energy as the model biofuel feedstock. And so with the naming or the designation of the species as a model species, there was a lot of research, influx in research with regards to looking at different things like improving specifically biomass yield of both upland and lowland cultivars, and also looking at other factors such as abiotic and biotic stresses that can affect switchgrass production. And again, keeping in mind that the most important trait or factor that we're looking at is biomass yield in the long term. So looking at how biomass yield is impacted by different factors, both environmental as well as disease and pest. And so for our purposes, with regards to Rutgers University as well as Cornell and Penn State, we've been really focused on breeding switchgrass for marginal land production, because going forward, that's going to be the most likely where switchgrass will be produced for bioenergy purposes, to avoid the food fuel debate, and also to breed for better disease resistance. And although there's been limited research on the effects of disease on switchgrass biomass yield, it's been shown in other warm season grasses that it can negatively impact yield overall. So moving right into project 1 that we've been working on for the past few years, we're basically looking at selecting and developing switchgrass for marginal lands, and in particular, we've been focusing on development for reclaimed mine lands. And so the real reason, or the main goal of this project, was established because we know that there are, several acres and thousands of acres of land, marginal lands, that are available for switchgrass production. But generally, when we develop cultivars of switchgrass or screen for improved germplasm, it's conducted on prime soils that are typically associated with experiment stations at universities. And so because we know that genotype by environment interactions influence switchgrass performance, which has been shown widely across a number of different locations within the United States, it can also affect biomass yield. And so in thinking about that and using switchgrass for marginal land production, it's not well known. It's not well known how the performance of switchgrass will be influenced by things like soil type. It's known that things such as temperature and moisture availability affect it, but it's not understood how selecting and breeding on prime soils will affect biomass yield in the long run. So with that, we developed the objective to determine whether we can screen, select, and identify switchgrass germplasm that can be used for cultivar development for improved yield on marginal soils, and whether that can be conducted on prime soils. So for this study, again, it was conducted in collaboration with Cornell University, and we were using 75 half-sib lines from both the Rutgers and the Cornell breeding pools that had developed specific switchgrass lines. We were using 3 separate locations. The location in Freehold, New Jersey is our research station. It's located south of the main university in New Brunswick, New Jersey, and that was considered to have a prime soil. The Ithaca, New York was associated with Cornell University, and again, that location had prime soil. And then finally, we were working in Phillipsburg, Pennsylvania, which is right outside of Penn State University, on a reclaimed mine land. The switchgrass lines that we were going to evaluate were started in a greenhouse in 2013, and then they were planted at all locations. So in particular, we had the Rutgers lines. They were planted in New Jersey and Pennsylvania, and then the Cornell lines were planted in New York and Pennsylvania. And so this is just basically showing you the locations where we were at. So again, in New Jersey, we're in a unique area in the transition zone where we can grow both upland and lowland ecotypes and have them do fairly well with regards to biomass yield. For Cornell, generally due to winter stresses, upland ecotypes tend to do better there. And again, at Phillipsburg, we were growing both upland and lowland ecotypes. So I wanted to focus on, basically give you an idea what the reclaimed mineland site looked like, because it was a pretty rough spot to plant these plants in. It was pretty disturbed, and as you can see, the soil here, prior to being used as a reclaimed mineland, it was a silt loam, but now that you can see that there are several rocks, it's very shaly. It's dry on the subsurface of the soil. And so at time of planting, this was in May 2013, you can see that the plants are fairly big that are being put into the field. But again, it's a pretty harsh environment for them to be planted in. And so when after planting in July, the time of planting in May and July, there was a very hot spell where it was very dry, and then there was also a frost. And so there was a lot of initial damage or kill to the plants that were put in the reclaimed mineland because of those initial conditions at time of establishment. So again, in going forward, it looks as though at time of establishment, what happens during that period is going to be very important for survival and ultimately looking at how much of your stand will survive. Another thing you'll in July is that you can see that there are a number of different weeds that have established in these planting areas. And so that's one of the #1 things that we were dealing with at the Myland site is weed management and making sure that those weeds weren't encroaching around the switchgrass plants, which took a number of different herbicide applications to control, but you can see from these sites that, for example, I can't get the arrow to work, sorry, but there are switchgrass plants that are planted in here and then are surrounded by weeds. And so this is just an example of a switchgrass plant and what it looked like following planting. So again, just to reiterate, we did see that a number of the plants did die once planted due to the environmental conditions immediately after planting, but we did have, fairly good survival for being planted in such a harsh environment. So just for those of you who are interested in what we did for management techniques, again, the switchgrass was planted in May 2013, and for weed management, there were several applications of herbicide that were made to control weeds, as well as any volunteers of switchgrass that may germinate from seeds. Nitrogen was also applied in June of 2014 and 2015 in order to supplement the soil. And so other than that, we were not applying any other pesticides such as fungicides, because one of the things, again, we wanted to look at was potential differences in disease resistance. So after the plants were established in 2013, we began taking a number of different measurements. They were taken first in 2014 and then repeated in 2015. Throughout their growing season, starting in around May, we were evaluating a number of different agronomic characteristics, such as vigor. So this is how vigorously the plants are growing, and it's based on not only plant height, but number of tillers as well. And this is a visual estimation based on a 1 to 9 scale, where 1 is not very vigorous and 9 is the most vigorous. Plant height was also measured, generally twice a year, one in the spring and then once closer to time of harvest. We also looked at maturity on a 1 to 5 scale, where 1 is the plants are still in a vegetative state and 5 is post-anthesis. This was measured a couple of times during the season, but mainly in the late summer. And then percent disease was also rated. And so depending upon what type of disease we saw in New Jersey and in Phillipsburg, it was mostly Anthracnose, which I'll talk about in the next project. And that was rated on a 1 to 100% disease scale. So because there were 75 lines in total, we decided to harvest a subset of those lines for biomass yield determination. And so those lines, 40 of the Rutgers lines and 39 of the Cornell lines, were selected based on vigor scores, and then they were harvested in 2014 and 2015 to determine biomass on those particular lines. And so those lines were harvested in both the prime sites, so the Rutgers lines were harvested in New Jersey and the Cornell lines were harvested in New York, and then they were also harvested in Phillipsburg at the reclaimed mineland site. So now we're going to move into the results for this project. So I'd first like to talk about, this might be a little bit tricky without having a pointer, I'll see what I can do here, but if we focus on, so basically what we're looking at here is a plot for our vigor scores. And so if we look at the New Jersey vigor score, so for the Rutgers lines, on the bottom part of this diagram, nope, that's not going to work. So if you look at the x-axis on the bottom of the diagram, you can see that there are negative numbers indicating where the plants performed poorly at our prime site, and there are also positive numbers, meaning they performed well at our prime site. In contrast, on the y-axis, we have the same sort of scale for our marginal site, which is the reclaimed mineland site in Phillipsburg. So the negative numbers indicate where the plants were performing poorly, and then the positive numbers were the plants that performed well at the mineland site. So what we did for selecting plants is we looked at overall performance based on vigor scores, and we basically took all of the plants, selected all of the plants in the top right quadrant, so meaning that those plants performed well at both locations. And we selected all of those for biomass yield determination. We also selected most of the plants that performed well at the mineland site, and we selected most of the plants that performed well at our prime site, and then took a couple that didn't perform well at either site. And we did that just so that we could get a range of performance to look at how the lines are actually doing at both of the locations. And although I didn't talk about the New York lines, we did the same thing for the lines that were from Cornell University. And so again, in total, that gave us 40 lines to harvest for the the Rutgers populations, and 39 lines to harvest for the Cornell population.
Let me quickly interrupt you. I changed the settings so that now people can see where your cursor is hovering above the screen at all times. So hopefully you can just use your cursor on your mouse to indicate the parts of the graphs that you'd like to showcase.
Okay, sure. So is that the hand? I'm seeing as a hand.
Yes.
Okay, perfect. Okay, that's great. Thank you, Sarah. That will make things easier. Okay, so what I'd like to do is I'm going to focus on biomass yield of the Rutgers lines in particular. I will talk briefly about the Cornell lines. So I'll set this graph up because the next few graphs are in similar format. On the x-axis, you have the switchgrass lines. And so for the most part, what you're looking at is just an arbitrary NJ number, which corresponds to a breeding line that was developed at Rutgers. But what you also see is that we have three standards. So we have Carthage, Cave and Rock, and Canlow. We included those at both the prime sites and at the marginal site. On the y-axis, you have kilograms dry weight per plant. And then within the graph, you have both upland, which are yellow, and then lowland, which are that rosy color. The bars on each of the lines on each of the bars represent an LSD. And so what we're looking at for biomass yield in 2014 here is from the prime site in New Jersey. And so overall, what you can see if you look at the trend, that there was significant variability in biomass yield depending upon the line itself. If we focus on specific lines, you'll see that overall looks as though some of the uplands had a little bit better performance compared to the lowlands, and in particular you can see that this NJ34 had significantly better biomass yield in comparison to a lot of the other lines that we were investigating. And also in comparison in particular to the three standards that we were looking at. And so this will be important to keep this NJ34 line in mind, and I'll point it out in the next slide. But you also see that there are some lowlands that had better performance, such as this NJ57, compared to some of the other lines that we were looking at. So if we move on now to biomass yield 2014, and now in particular we're looking at yields from the reclaimed mineland site, we're looking at the same scale and the same lines. And one of the major things that you should notice is that biomass yield is significantly lower in comparison to the prime site. So yields were much lower at the marginal site, which was to be expected. We could visually see this in the plants when we were evaluating them. And you'll see that the three standards are, their yields are lower in comparison to a lot of the lines that we were investigating. But if you remember that NJ34 line from the previous slide, you can see that it is no longer the best performing line in comparison to all the other lines that we harvested. We see now that this NJ30, which is an upland, now has better yield than a lot of the lines that we harvested in 2014. So with only a year's worth of data, though, this kind of indicated that, again, performance is going to vary depending upon the environment, which is kind of an indication that, the screening and selection is site-specific for developing cultivars for marginal sites. So now if we move into 2015, one of the things I want to point out first off is that we saw at our prime site in New Jersey that biomass yield was significantly lower in 2015 compared to 2014. And so there's a number of different factors, which I'll discuss a little bit about, but one of the things that we're thinking why we had reduced yields in these lines is because of the time that they were harvested. They were harvested earlier in October, when they were still somewhat green, and it's recommended that you wait until the biomass is completely brown until harvest time. So this may have affected the recuperative capacity of the plant to grow back in 2015 from the harvest in 2014. What you'll also notice is that we see a shift in trends with regards to performance for some of these lines. So again, 2014, we saw that this NJ34 Upland line was one of the best performers, and now it's one of the middle performers, but we see that this Lowland and NJ36 has better yield than most of the other lines that we were evaluating in 2015. And we also see that this NJ6 is one of the top performers as well. And so I'll discuss briefly about what some of the biological factors are that may have contributed to this. But in New Jersey, we saw that in 2015, was very hot and it was very dry during this growing season. And so it's not surprising that we see that the lowland, which is more adapted to warmer climates, has better performance than some of the other uplands that we were evaluating. In contrast, we saw a significant increase in yield at our marginal site in 2015. And again, you see that there's variability in performance depending upon the line, with some of these NJ47, which is a lowland, and NJ30, which is an upland, having better performance than a lot of the other lines that we harvested that year. And so again, we're seeing differences not only between sites, but also between years. And so when we were looking to evaluate what's causing these differences, which you can see here, this is the New Jersey nursery in 2014, and so you can see how large those plants are. We were using a tractor to hold a small scale up, and this is how we were weighing the plants for biomass yield determination. You can see that in 2015, these plants are much smaller, and this is prior to harvest, obviously, but much smaller compared to 2014. Now, if we look at the nursery in 2014 in Pennsylvania, you can see that those plants are pretty small. And so we actually were able to harvest all of these by hand, which was relatively easy in 2014. In comparison to 2015, you can see that these plants are much larger and they have many more stems than in the previous year. So the flip in this trend for biomass yield production, we think is primarily associated with the amount of rainfall that each of the sites received. So if we look at our Freehold, New Jersey site and we compare that to our reclaimed mineland site, you'll see that in 2014, not a big difference between rainfall in June, but in July and August, there was more. Am I erasing that? No, okay. So there was more rainfall at Freehold in comparison to Phillipsburg. And so we think that, again, those plants did well in 2014 at our prime site, and it may be associated with that rainfall. And then if we look in 2015, that trend is different. So we see that in June, there was a significant amount of rainfall in Phillipsburg in comparison to Freehold, and that trend held true throughout the growing seasons of July and August. So again, in looking at the performance on marginal sites, it looks as though rainfall is going to be one of the most important factors that's going to contribute to biomass yields on a yearly basis. So briefly, just to look at the Cornell lines, I just wanted to show you what the trends that they were seeing. And again, it was very similar to the performance of the Rutgers populations, where we saw biomass yield was significantly greater in Ithaca, New York, compared to the reclaimed mineland site in Phillipsburg. We also saw that performance varied depending upon the line and the ecotype, but we saw that performance on the prime sites wasn't necessarily reflected on the marginal sites. And for the the Cornell populations, this held true also in 2015. So although they saw that biomass yield was slightly higher at the reclaimed mineland site for 2015, it was still higher at the prime site. And so again, Ithaca had similar rainfall amounts for the Phillipsburg, Pennsylvania site, and so that might be the reason why you're still seeing higher biomass yields at the prime site compared to the marginal sites. One of the other things that we wanted to do after our first biomass yield harvest was to determine whether we actually captured the best performing lines. And so what we wanted to look at was whether there was a correlation between biomass and vigor depending upon the line and the location. So when we looked at vigor, which has been shown to be a good predictor of biomass yield, compared to biomass in New York, there was a significant correlation not only for Cornell lines, but also for the Rutgers lines. And this held true for the prime sites as well as for the marginal sites. And so for us, that meant that going forward, using vigor is potentially going to help increase the efficiency that we can screen germplasm and select germplasm for increased biomass yield. And the other thing that was interesting is when we looked at the correlation between biomass at Pennsylvania and New York and biomass in Pennsylvania versus the Rutgers lines, you see that those are not significant, and the same for vigor. So what this means is that it's site specific. So when you screen germplasm, it has to be done at the location where those lines are going to be grown. When we look at these correlations again with the 2015 data, we see a similar trend for the different locations. But one interesting thing that we saw shift, which can be explained by the differences in biomass yield, is that biomass at the prime and the marginal site for the Rutgers lines and the same for the vigor, those were now significantly correlated. So again, the change in that relationship is most likely associated with the fact that we saw a shift in the trend where biomass yield was higher at the marginal site compared to the prime site in 2015. So again, this ties back into the genotype by environment interactions that we commonly see with switchgrass and its performance, and really says that, screening has to be done over multiple locations as well as multiple years to accurately capture the performance of a particular line before advancing it for cultivar development. And so this is a picture from last year on our annual NEWBio meeting. We had a tour which went to the reclaimed mineland, and you can see that this was the summer 2016. Our plants are still doing really well there at that reclaimed mineland site. So again, this would be their fourth year now. This in 2016, it was their third year. And so again, it shows that it may take a couple extra years for those plants to get established and to grow well on a reclaimed mineland site. So again, it's something to keep in mind even when evaluating the plant material or for production purposes. So to summarize this first project, which I mentioned this already, but the performance at the marginal site compared to the prime site definitely depended upon the switchgrass line, and not only the line, but the ecotype and also the year. So it's going to be the performance of these lines are going to be significantly influenced by factors such as temperature and mainly rainfall. We also found that we can use vigor to accurately select for improved biomass yield, but again, screening has to be done at multiple locations, including marginal lands, especially if that's where those switchgrass lines are going to be produced. And lastly, we selected plants for the development of new switchgrass cultivars based on the data that we collected in 2014 and 2015. And so again, this is looking at biomass in both New Jersey and New York lines with the same scale as we used for selecting the plants based on vigor, with the positive performance or the negative performance depending upon whether it's a marginal site, which is on the y-axis, or if it's at the prime site. And so the way we selected plants for crossing blocks or for developing cultivars is that, again, we wanted to take lines that could perform well at both locations, was our primary primary selection criteria. And so based on that criteria, we selected 15 lowlands and 15 uplands for crossing blocks, and that was from both prime land as well as from the marginal site. And so in order to make sure that we were establishing isolated crossing blocks, the Rutgers group took all of the lowland plants and the Cornell group took all of the upland plants. And so here these plants were dug up from each location and then planted in the spring of 2016. And so here you're looking, this was basically immediately after transplanting these into a crossing block. And these were the lowlands that we were planted in Freehold, and these were the uplands that were planted in Ithaca. We also did the same thing with plants that were selected from the prime sites. So again, the lowland plants were planted in Freehold and the upland plants were planted in Ithaca, New York. And so last year, again, we had some pretty hot and dry conditions in New Jersey as well as in New York. And so in order to promote seed production in both the prime and the marginal crossing blocks, we did water on several occasions and then applied fertility to the blocks once during the summer in order, as I said, to expedite the seed production so that we could harvest seed in the fall of 2016. So going forward with this project, we have a manuscript that's in the process of being written and will be submitted to Bioenergy Research that will recap the 2 years of this project. Last fall, as I had mentioned, the seed from the crossing blocks was harvested and it's currently being prepared for future use. So we have a potential grant that would be funded that we may be able to use to screen those plants that were developed from those crossing blocks. And then going forward this year, we may select additional plants based on performance of this year and how they look in the field, particularly at the marginal site in Phillipsburg, Pennsylvania. So the next project, Project 2, that we're moving on to is focusing on developing switchgrass lines that have better disease resistance, in particular to Anthracnose, which is Colletotrichum navitas. And to give you just a brief overview, Anthracnose on switchgrass was originally thought to be caused by a different Colletotrichum species, but until 2009, when Dr. Bonos was working with Dr. Crouch and others at Rutgers University, they discovered that it was actually a unique Colletotrichum species, which ultimately was named Colletotrichum navitas, where navitas means energy in Latin. And so in particular, what you see with Anthracnose is that it usually infects the leaf tissue first, and then you could also see infection on the stem tissue. And this happens during hot and humid weather. And you'll see that you have these lesions on the leaf tissue, which can also happen on the stem tissue. Initially, they start out as yellow chlorotic lesions, and then you'll see this brown circular ring, and then ultimately it will become bleached within the leaf tissue. And so, although there's not a lot of research on the impacts of Anthracnose on biomass yield in switchgrass, it's been shown that Anthracnose can reduce yield in other warm season grasses, primarily associated with the loss of photosynthetic tissue during the growing season. So what we see with Anthracnose, the signs usually are these setae, and we don't usually look for these in the field. This is something that you could bring in and look under a microscope in the lab. What we typically see for disease development is the symptom, again, as I had just described, are these lesions on the leaf blade, which can also happen on the stem, that turn out this black color that becomes chlorotic, and then eventually the center becomes bleached. And this can occupy, on some of, depending upon the cultivar, 100% of the leaf tissue. So because there's limited information on how Anthracnose influences biomass yield, and when looking at production for bioenergy purposes, more than likely this switchgrass is going to be grown as a monoculture, which can thereby increase the amount of disease epidemics, both the severity and longevity. We really needed to look at how that Anthracnose can affect both the quantity, so the amount of biomass, as well as the quality of the biomass, in turn, which could impact how it's processed for bioenergy purposes. So our three main objectives for this study were, one, to determine whether Anthracnose significantly reduces biomass yield of switchgrass, and look at how anthracnose affects different cultivars, including both upland and lowland ecotypes, and then ultimately look at if there's any impact of anthracnose on lignocellulosic characteristics. So it's been shown in other plant species that when the plant is being infected by some sort of pathogen, that there's a response where you have an increase in lignin. And so if that's the case, that could ultimately, as I had said before, impact how it's processed for bioenergy purposes. So in 2013, 16 cultivars or populations were seeded by hand, and that included both upland ecotypes, which are listed here, and lowland ecotypes. So we have some common cultivars that are available. Carthage C2 and Timber C2 are lines that were developed at Rutgers and cycled through the breeding program a couple of times. And then the New York lines here were from Cornell University. So again, this was a collaborative project with Cornell University, but only conducted in New Jersey. For our treatments, we were applying fungicides to only half of the plot area of the seeded plot area. And so this is a picture of our research area. And so this entire plot would be one cultivar. And so we were spraying half of this area with fungicide and leaving the remainder of the plot untreated. So in 2014 and 2015, we were using a combination of Daconel, Alliette, and Heritage in rotation. And so we made a number of different applications in both of those years. And based on the results, we decided before the growing season of 2016 that we needed to try different fungicides because we weren't seeing differences based on treated and untreated plots. So in 2016, we made 8 applications of Daconel plus Valista, and we rotated that with Mirage and Alliette. And these fungicides were based on recommendations from the plant pathologists within our department. And although there are no fungicides listed for switchgrass, we were basing it off of their efficacy on other cool and warm season grasses. And so again, just to reiterate, the non-treated plots did not receive any pesticide applications during the growing season. Throughout the season, basically starting in July or late June, I was rating percent anthracnose on a visual basis from 0 to 100%. And then there were other measurements as well, as maturity or percent cover, but I'm going to focus on anthracnose as well as biomass yield. And so for the plots, we determined biomass yield in October of 2014, 2015, and we also determined it in 2016, which I did not add there. So we were harvesting a known area of switchgrass from each of the treated and the non-treated plots using a sickle bar mower. So this was relatively easy to do, where we took this known swath. One of our technicians would go through, and then we would come through and rake up all of the material that was harvested and then put it in a bag, and we would weigh that after drying for biomass yield determination. And then this is what it would ultimately look like after the harvesting. After the samples were dried, they were ground and they were used to determine biomass composition using NIRS. So now moving on to the results for this study. And again, I'm focusing particularly on susceptibility to anthracnose based on a visual rating, and then later we'll talk about biomass yield. So I'll just set this graph up because the next view will look like this. On the y-axis, you have percent anthracnose on 0 to 100%. And on the bottom, on the x-axis, you have the cultivar or population name. The yellow represent the upland ecotypes, and the pink color represent the lowland ecotypes. So again, these cultivars are listed from most disease to least amount of disease. And this again is for 2014. And so what we saw again was a range in responses to anthracnose, but what you'll notice is that there are no differences between fungicide treated or not treated. So that data is pooled together because there were no visual differences between the two plots in September of 2014. And again, we may have seen differences during the growing season, but the most important date for looking at the differences would be right before biomass yield harvest. So we see that a lot of the uplands had more disease in comparison to the lowlands, and in particular, we saw that Forestburg was the most susceptible to anthracnose infection. And again, at certain points, we saw even more, greater than 80% disease, but this cultivar in particular got the disease earlier and faster in comparison to all the other cultivars that we were evaluating. And we saw some of them, like Timber C2, had a limited amount of disease in comparison to some of the upland ecotypes. So keeping the same order in 2015, we see that the response was a little bit more variable. We still saw that Forestburg had the most amount of disease compared to all of the other cultivars evaluated, but we saw the performance of some of the other lines, such as this NY13309. This was a little bit higher in 2015, where Carthage C2 had a little bit lower disease severity. But again, we did not see that there were any differences between fungicide treated and fungicide not treated plots. And again, this visual rating was taken right before harvesting. So this was, again, one of the reasons, the main reason that we decided to try different fungicide rotations for 2016. So although we are seeing differences between the cultivars, so you can see this is Carthage in comparison to Forestburg, we're not seeing significant differences between the treated and not treated treatments. However, in using the new chemical compositions in 2016, and we did have a couple of extra sprays during that growing season, we did see a difference between treated and non-treated plots. So again, the upland not treated is this yellow, and then the fungicide treated has the lines, and then the lowlands are that pink color not treated, and then the lowlands with the lines were fungicide treated. And this trend that we're seeing for this October 16th rating held true throughout the summer. And you'll see that, again, Forestburg, along with Blackwell now in 2016, had the highest amount of disease in comparison to a lot of the other lowlands. But what you see is that fungicide did help in reduce the disease severity on all of these cultivars that we were evaluating. So for us, this was promising for looking at biomass yield. And again, this is just visually what we were seeing in the field. And you can see that the not treated here has more senesced tissue. It's more brown in comparison to the fungicide treated, which has this more green tissue overall for the entire plot. And again, we were seeing this for all of the cultivars, even for Forestburg, which seems to be the most susceptible to anthracnose infection. So now we're moving back to 2014, but looking at biomass yield. And so if you remember back to the anthracnose, we didn't see any differences between the fungicide treated and not treated plots, and the same held true for when we looked at overall biomass yields. So we're looking at megagrams dry weight per hectare on the y-axis, and again, you're seeing the cultivar population name on the x-axis. We see that Forestburg, which had the most disease, has the lowest yield, and that may not be a function of the anthracnose. It was overall one of the shorter cultivars that we were working with, so that may be partially an artifact of its overall traits in general, not just purely associated with the fact that it had more anthracnose disease. But we do see that there are some higher yielding cultivars that we were evaluating, in particular something like this Carthage C2, which I said was cycled through the Rutgers breeding program. So we do see variability in biomass yield, which is expected, and we also saw that trend in 2015, although it was higher in 2015 compared to 2014. We still did not see a difference between fungicide treated and not treated plots. But again, you see that Forestburg is the lowest yielding cultivar in comparison to all of the other cultivars that we were evaluating. So again, we had hopes based on the visual differences we were seeing in the cultivars for biomass yield in 2016. But ultimately, when we harvested the biomass yield, dried it, and weighed it, we did not see that there were differences between the two treatments. Now, although I have this broken down by whether it was not treated or fungicide treated, I just wanted to show you that how closely the biomass yields were for a lot of the cultivars that we were looking at. So again, although we're controlling the disease somewhat, we're still seeing a significant amount of disease in the plant tissue. It does not appear to be affecting the biomass yield in general. And then when we went ahead and analyzed the plant tissue, and I only have the data from 2014 here presented because we saw similar results in 2015, we did not see that whether it was fungicide treated or not treated, that did not have a significant impact on composition. And then there does not seem to be a correlation between anthracnose, severity of anthracnose, and then biomass composition in general. So we did see differences in total cell wall content, but like I said before, that's probably not associated with anthracnose infection or severity of what the cultivar was looked like. So with this study, it's slightly frustrating because we're seeing that we are getting some efficacy of the fungicides and they have some impact on controlling fungicides, but we're not seeing close to 100% control on the switchgrass cultivars. And so that's really hard in looking at the data in the field and then ultimately making decisions on whether anthracnose is impacting switchgrass biomass composition or biomass yields in general. So there may need to be some subsequent research in controlled environment chambers looking at fungicide efficacy to get 100% control and then translating that into the field and looking at that. And as I had mentioned before, when we actually look at anthracnose severity and how the cultivar is affected, we did see that Forestburg was the lowest yielding cultivar and had the highest percentage of disease. There was a significant correlation between those two factors, but it may be an artifact, like I said before, just of Forestburg and the fact that it is a lower growing, less vigorous cultivar compared to some of the other cultivars we were evaluating. And so again, the relationship between anthracnose severity and biomass yield was not consistent across all of the cultivars that we were evaluating. And then lastly, when we looked at biomass composition, it does not appear to be affected by anthracnose severity, the amount of disease that's impacting the plant. And so we really need to actually take a look at the changes in plants that are maybe not infected versus infected. So this would require, again, doing a controlled study and inoculating the plants. But if doing it in the field, we need to get better control of the anthracnose than we were with the fungicides that we were using. So again, it comes back to finding which fungicides are going to give us the best control in a field setting. So going forward, we have made 2 additional fungicide applications to the field following harvest in 2016. There still hasn't been a decision made about how we're going to proceed for the growing season of 2017. The funding for the NEWBio grant is coming to an end, and again, so there's not enough to conduct this study for another year, but there is potential that we may continue doing another year of evaluation to see if we can get any differences between the fungicide treatments and also looking at how that impacts biomass yield. So with that, I would just like to thank or acknowledge all of those who have been involved in the biomass harvest, which is very time consuming and can take in. So I appreciate all of the help from those at Rutgers University and Cornell University, as well as from Penn State University. I'd also like to thank NEWBio for funding my position over the past 3 years. And with that, I apologize for some of the technical issues that we had throughout the presentation, but With that, I'd like to thank you, and I'd be willing to take any questions in the chat box if anyone has any.
Thanks so
and
much.
so, sorry, before we ask questions, I'll just give you kind of an idea. So this is one of our crossing blocks from the Marginal land study, and this is how I was harvesting them. So they were fairly large. They grew very quickly within the growing season of 2016. So I was zip tying the top to harvest the seed, and then harvesting the seed by hand, and then drying them prior to thrashing. And then these were my helpers, Charlie and Colt. So with that, I would take any questions.
Good scientific practice to acknowledge your contributing dogs on every project. So I'm glad to see you've done that, Lindsey. As Lindsey said, if you have any questions for her, type them into the chat pod and I can pitch those questions to her and we can have a conversation about this. We have about 5 minutes left until 2 p.m., but if any of you are willing to stick around for some really burning questions, we can go a little bit long today. So Lindsey, let me just start, since we just came fresh off the Anthracnose discussion. It's interesting that there aren't listed fungicides specifically for switchgrass, even though it's a fairly well-established crop that's known to some commercial producers. When commercial producers face the question about treatment, is treatment even common for switchgrass fields that have anthracnose infections? What fungicides do they tend to use if they do treat?
So I don't know 100% if growers that are growing switchgrass on a large scale are treating with fungicides. My guess would be no. One, because again, we're trying to promote switchgrass or use switchgrass as a lower input crop. And so from a sustainability and environmental perspective, it wouldn't be practical to do that, but also from a monetary standpoint, to spray large areas of switchgrass would cost a lot of money from the fungicide, and then also utilizing the equipment to spray the fungicide and also take up a lot of time. So I think especially that there's not a lot of research correlating biomass yield and anthracnose together. From a fiscal standpoint, I don't think that it's worth spraying, but I don't know. I don't think that there are people out there spraying currently because of those reasons.
Yeah, you're right. Especially because it's not clear that there is that drop in yield or in quality of the lignocellulosic material. So your presentation, both of the aspects of your presentation underscore the importance of site factors in cultivar performance. So let's consider site factors for anthracnose quickly. So you mentioned things like hot, humid weather. Obviously, moisture is a big part of that. So some of what drives farmland marginality in this region is site wetness. Do you see any evidence for increased anthracnose pressure in more poorly drained sites for a given cultivar? Is that something you've looked at or something you have an interest in?
So we don't have any poorly drained sites that I, in particular, have been looking at. Our reclaimed mine site in Phillipsburg would tend to be more on the dry side from a soil standpoint. But from a humidity standpoint, we did see that the plants in Phillipsburg got anthracnose both in 2014, 2015, and we did see some in 2016. I wouldn't say that it was more or less severe than our prime site in New Jersey. I would think that they were about equal. I think it's going to have to do, the severity of the epidemic is going to be related to the overall health of the plant. So again, if it's growing in a soil that is nutrient poor and the plant is under drought stress or some sort of heat stress, it may be more susceptible to the disease. And also how much switchgrass is actually being grown. So we have other sites of where inoculum can come in at our prime site. And so there's more of a potential to have greater outbreaks, greater epidemics of anthracnose when there's more plant material. But again, it's also going to be influenced by overall health of the plant itself. So from a soil wetness standpoint, it would depend upon whether the plants were exposed to flooding stress for prolonged periods of time. Yes, that might induce a little bit more disease, but it's going to be associated more with how humid and how hot the conditions are and for how long those conditions persist.
Thanks. Yeah, and your point about compounding stressors is an important one, for any cropping system or any vegetation management system. All right, I have one more question. I'm going to push you for practicality reasons. So brass tacks, when we talk about improving cultivars, what would you say is a typical timeline for cultivar development and release for commercial planting? I think you've underscored the really important point of needing to do this work over multiple sites and multiple years to gather, correct data and sufficient data to make some conclusions about what works where. I mean, are we talking like 10 years from when it's tried to when it's released for commercial development?
Yeah, so I just had to consult with Stacy a little bit here. So because this is only my third year working in the switchgrass breeding program, I wasn't really 100% sure, but Stacy's saying around 9 years, and actually said that Calvin probably has a pretty good idea of that too for the development of new cultivars. But it's really going to surround or be influenced by the stability of performance of that cultivar. So again, if we need to screen on multiple locations and look at the performance of biomass yield and how that line is performing not only on marginal sites, if we're moving forward with promoting those as grounds for large production switchgrass venues, and then look at the performance on prime soils as well. So typically between 9 to 10 years from initial screening of the germplasm to the release of the cultivar, yes, would probably be typical.
Given the length of managed vegetative systems, it's still a fairly nimble turnaround if you think about it. Thanks for that information. Since you have Stacy there with you, is there anything else about this topic that you or she would like to weigh in on? We're running up against 2 o'clock, so I guess there is still room if anybody wants to ask another question to type it in the chat pod. But in the meantime, are there sort of final thoughts you want to leave us with today?
I mean, overall, in general, it's been a great opportunity for me to work with the NEWBio consortium to get to know all of the different researchers and all the different components of the consortium itself and what goes into not only from a breeding perspective, because as breeders we focus heavily on the plant material, but also on the policy aspect and looking at it from a feasibility standpoint. From our projects in particular, I think we've had some really interesting outcomes with regards to the marginal land screening, and I hope going forward that we're able to continue that and really screen the the germplasm that we've created from this grant. So we're hoping that we can do that and continue our work in Phillipsburg and continue to have updates via fact sheets or published manuscripts for people to get the information out there for people to read.
Thank you so much, Lindsey. With that, I think we'll conclude today's presentation. There will be a recorded and uploaded version of this presentation for more wide sharing available on our website either later today or tomorrow, and you can also find that linked through our Twitter feed. I want to thank everyone for joining us today, and thank you again, Lindsey and Stacy, for your contributions here.
You're welcome. Thanks, everybody.